Hello, in Mr. Sharp's class this year, we used venom and added anti-venom to the venom to see how fast it would activate and if hot or cold temperatures would affect the time for the anti-venom to work and to save a patient who has been infected with venom. Second, in Mr. Sharp's class, we did a crime scene investigation where the body of Anna Garcia was found with clues leading to the reason of why she died. Here she was found, and basically this helped us to learn how to investigate, to go through a crime scene,
is taking his
etc.
fingerprint using pencil graphite and tape. And basically, we did this to learn how easy it is to take fingerprints, what fingerprints look like, and what ours are like.
Hi, this is Alec Foles, and in the blood splatter lab, we dropped blood from different heights to see how far it would go, and this would lead us to details about Anna Garcia's crime scene.
In the strawberry DNA extraction lab, we extracted DNA from a strawberry. In order to do this, we followed this procedure. The reason we use a strawberry's DNA is because the concept is the same for human DNA, but instead you use saliva. And also, the DNA of a strawberry looks very similar to a human's DNA.
Heart dissection lab, we dissected a sheep heart. We took red and blue string and put them through the aorta, ventricles, atriums, and the vena cava. The end goal was to be able to hold the heart up with just the strings going throughout the heart.
During the nutrition lab, we studied food labels and researched how to read them. We also learned that you can tell if a food is healthy by reading the nutrients on the nutrition label.
In the phlebotomy lab, we drew blood and learned how to do it safely. Then we placed it into a centrifuge to learn how they worked and how they removed cells from each other.
During the gram-staining lab, we observed how different chemicals show if a bacteria is gram-positive or gram-negative. We applied crystal violet first and flood the slide for a minute. After the minute is done, we rinse it with water. Next, we apply the slide with iodine for a minute. After the minute is done, we put water on it. We rinse the slide with water. Then we put alcohol on the slide for 15 seconds. After the 15 seconds is up, we rinse the slide with water. Finally, put saffron on the slide for a minute and rinse the slide with water. Then we wait for the slide to dry. Then we can see if the bacteria is gram positive or gram negative. Gram negative is pink and gram positive is purple. Perfect. During the moldy bread lab, we collected fungi around the school and tried growing it in a bag with bread. First, we collected the fungi with a swab and put the fungi in a bag. The whole bread is infected with the fungi. It goes through the bread with the hyphae, which cannot be seen. There are different types of funguses. Some are bad and some are good. In the end, fungus grew on our bread from our experiment. We collected the fungi from a gym locker room door.
That's perfect.